In this talk, I'll be describing how we use interferometry and coherence and speckle and laser ranging to use motion as a way of measuring properties of life. And I'll say a lot about intracellular motion and Doppler light scattering, and then I'll describe this imaging technique that we've been developing over many years now called biodynamic imaging. And there are a range of applications I'll talk about: chemosensitivity testing, mostly for cancer, then quantitative phase imaging, and also living tissue sentinels. So this is the parameter space that we're dealing with. And so what I'm showing here is velocity or speed in microns per second as a function of the size of some intracellular component. And so we're going from small sizes to large sizes. So then on the small size, you're looking at organelles, and the large size, you're looking at the whole size of the cell. And the speeds are actually spanning from about tens of microns per second down to tens of nanometers per second. So we're spanning about 3 orders of magnitude on the vertical here, but not quite as wide a dynamic range on the size. And so the bottom line is that small things move fast, large things move slowly. If this sounds familiar, it's pretty much a statement of Stokes' law of drag, except this is now Stokes' law for active transport inside of cellular media, because what's really happening is that velocities are essentially all at their terminal velocity. This is life at low Reynolds number. And so velocities have an immediate response to forces that are applied. Inside the cell, most forces are in the range of about of piconewtons, maybe up to tens of piconewtons. This is very standard, whether you're looking at a molecular motor or whether you're looking at the cytoskeletal restructuring. The forces involved are about a piconewton. And then you just have to consider the viscosity. Now, the viscosity inside the cell, it's a very crowded environment. And so it turns out the viscosity is scale dependent, but very roughly, there's a characteristic scale of about a micron, and the viscosity is roughly a Pascal second for objects that are around 1 micron. But there is a size-dependent viscosity. You put that into this generalized Stokes law for active transport, and you more or less get this line here with a lot of variability depending on what process. But these are lots of different active processes that are going on inside the cell. But what I really want you to focus on is this velocity scale. So tens of microns per second at the highest speed, down to tens of nanometers per second for the slowest speeds. All right, so everything going on inside the cell is active non-thermal transport. Now, it may be a random walk, but it's not thermal. It's driven by ATP. It's the hydrolysis of ATP to ADP, and that has a hydrolysis-free or Gibbs-free energy of 0.32 eV. And so this, for instance, is what's driving molecular motors. So molecular motors will be dragging a cargo along highways. And so the highways are the cytoskeleton. The cargo will be organelles or vesicles. And so they're carried along by these molecular motors. And if you look at this as a random walk, and these really are random walks, but they're highly persistent random walks. And you can define effective diffusion coefficients for this. And they're really defined by persistence times, that's the T0, and by average speeds, which are really set by the maximum rate of molecular motors. And so you can figure out what the effective temperature might be if this were Brownian or thermal motion, and it's thousands of Kelvin. And what's interesting is that if you also convert the hydrolysis energy to an effective temperature, it's also in the range of thousands of Kelvin. So it turns out that life is actually pretty efficient in using these energetic molecules for driving motion. And so it's a very highly efficient use of energy to drive this very active transport going on inside of cells. All right, so if things are moving, and then if you shine light on them and the light scatters, then there will be Doppler shifts involved. And so Doppler introduced his grand idea in 1842. He actually introduced it to a lot of ridicule. In 1842, nothing could move fast enough to actually experience the Doppler shift in sound. And so he was really proposing this as a phenomenon, but it was very hard to actually demonstrate it. And in fact, many people thought it was just plain wrong. And they held a trial. The National Academy in Vienna held a trial. They pretty much put Doppler, or at least his effect, on trial. And they found that it was nonsense. And so he was basically drummed out of Vienna, and he went off to Italy, where he died of tuberculosis. And so it's kind of a sad story for Doppler. But his champions continued supporting the idea. Ernst Mach was one of the main ones. And he demonstrated for the first time an apparatus that did demonstrate the Doppler effect. And then finally, trains got fast enough in the late 50s or 60s that you could actually hear the Doppler effect. If there were, in fact, the guy who did this actually put a brass band on the back of a train, and so then they had people with perfect pitch, and so they would listen to the pitch of the instruments as it went by, and so then that was the first demonstration of the acoustic Doppler effect. So I'll be talking about the optical Doppler effect, and so for instance, if I have a mirror which is moving in this direction, a photon is coming in from the left, and it reflects off the mirror, it goes back off to the left, and this will have a Doppler shift associated with it, and you can look at this as two different frames. There's actually a mirror frame, and that's when the photon is coming in. And then there's a lab frame, which is when the photon is coming back out. And so in the mirror frame, as far as it's concerned, the source was moving away when it emitted the photon. And so then that's a redshift. And so this is the relativistic formula for the redshift. And then after it reflects, you go into the lab frame, and so in this case, this is the receiver, and the receiver sees the mirror receding with that velocity. And so you actually pick up a factor of 2. And this is relativistic, although in the extreme non-relativistic limit, and so you do pick up this factor of beta, which is V over C, but you also pick up that factor of 2. And this is then for backscattering. So in backscattering, you have this factor of 2 of beta on the fundamental frequency, and then this is the change in the frequency, or what we'll call the Doppler frequency, because what we're going to be measuring are actually the Doppler beats, and the beats actually have this frequency here. I just want to put a scale on this, and so I mentioned that with intracellular motion, that we're looking between 10 microns per second down to 10 nanometers per second. But just a very convenient rule of thumb is that 1 micron per second is in backscatter, in near infrared, is a Doppler beat frequency of 3 hertz. And that is quite easy to measure. You can measure that with cameras and with very reasonable frame rates on cameras. All right, so since we're talking about living systems, we also have to talk about coherent speckle. And so one way of thinking about speckle is that you've got a laser beam coming in and hitting a heterogeneous target. There's lots of scatterers. And there's a lot of rays. So this is actually a physical optics, sort of geometric optics way of looking at it, but you have all these scattered rays coming off these particles. And so there's a dispersion in your K, which in the far field actually turns into this pattern here, which is a speckle pattern. And so speckle patterns will have characteristic sizes, and the sizes are set by the emission areas that you're exciting, and there's propagation distances, and there's also the wavelength of the light which is involved. And so you can actually calculate, given an excitation geometry and a propagation distance, what the average size of these speckles are. And they can be, for a typical experiment, let's say, something in the range of tens of microns. And so again, something very easy to capture onto a camera. All right, so again, we're talking about life. Life is moving. Everything about life, as I said, is motion. And so if you have speckle, because you're shining light into a heterogeneous system, it means that it will be dynamic. And so this is an example. This is real data. This is on a camera, and it's false color coded, but just on intensity. But you can see that it's highly dynamic. And so the speckle looks basically like noise. It's not noise. What you're looking at are the Doppler beat frequencies of all types of processes. So when you shine light on living tissue, everything's happening all at once. And so all those Doppler frequencies are getting together and beating against each other, and that's producing this dynamic speckle. So what we'll do with spectroscopy is we'll take a pixel, and then that pixel will have a time series associated with it. And so once you have this noisy time series, you can do one of two things. You can either look at its autocorrelation, or you can do a Fourier transform. Now, it turns out that under sufficient conditions, these are actually related by the Wiener-Khinchin theorem. And so the power spectrum is just the Fourier transform of the correlation function, and vice versa. We like to work in frequency because frequency is a very natural way of thinking in terms of the speeds going on inside a living tissue. And so we'll be working with power spectra, but in fact we could also be working with autocorrelation functions. Well, now we'll get into some of the details of Doppler light scattering, and that introduces this idea of something known as a Doppler number. It's a dimensionless number that tells you whether you're in a regime where there's long persistence or whether you're in a regime where it's basically like a Brownian random walk. So I mentioned that we're looking at Doppler beats, and so these are, let's say, examples of Doppler beats, and so then you'll get these random time reversals. In part, that's because most motion in life is quasi one-dimensional, and so things will actually change direction more likely than they're changing or changing the sign along an axis rather than changing into a new orientation. And so what you get is lifetime broadening. And so if you have a sinusoid, which then goes into these random time reversals, you'll actually get this peak here, right? So that's this dashed red curve. That is a lifetime broadened Doppler signal. And so you actually see the Doppler peak in that case. Now, you can define a Doppler number as being the Doppler frequency shift, so 2 pi f sub Doppler times a persistence time, or it's also the scattering k vector, q, times the persistence length. And so that's a dimensionless, right? So it's either omega t or ql, it's dimensionless, and this is the Doppler number. And they are equivalent, and you can see the equivalent actually right here. And it turns out that if your Doppler number is less than about 0.3, that puts you into a Wiener regime. I say Brownian but Brownian really is like thermal, and so I want to avoid that. So I actually want to call this more a Wiener process, which is a process that has derivatives nowhere, and so there's no persistence in the extreme Wiener limit. But then also, if the Doppler number is greater than 3, then you're in the Doppler regime, and that's where you have long persistence. And just as a rule of thumb, so if the Doppler number is 1, then the persistence length just has to be equal to the reduced wavelength, which it has a 4 pi. This 4 comes from the factor of 2 from the backscatter. That's the refractive index. It turns out that the persistence length only needs to be comparable to about 50 nanometers to be in the crossover between the Wiener and the Doppler. And so that's actually an easy condition to hit. And so I do want to also point out that if you have a Doppler number of 1, that you've more or less lost your Doppler peak. It's so lifetime broadened that it becomes an edge. And then if the Doppler number is very small, then you actually go over to this diffusive structure in this dashed blue here. And so then that would be like Brownian motion if it were thermal. One more just detail is that, in fact, in tissue, the motion is isotropic. So this Doppler peak, the dashed red, was for collinear motion to the q vector, but the q vector and the velocity are have any orientation, and so you have to do that angular average, and it converts the Doppler peak to a Doppler edge. And so here at a Doppler number of 10, you have a Doppler edge. For Doppler number of 1, you get this knee, and then for 0.1, you get this strong diffusive structure, and then that would be the typical dynamic light scattering signature that people see with thermal Brownian motion. So what's our Doppler number inside a living tissue? Well, it turns out that, there's hundreds of papers written on this, and so it's not so much that we're detecting speeds and motions for the first time. In fact, we're building on a vast literature of live cell microscopy, and so you can go in, and this is just a small sampling, but lots of different processes. Lots of papers written on the same processes, but what they all pretty much agree on is that if you consider the persistence length or the persistence times and the speeds, all of these processes, all these active processes inside, or I should say almost all these active processes inside a living tissue is in the Doppler regime. So your Doppler number is greater than 3 and possibly like around 10 or even higher. Now, of course, there's distribution of speeds, and so even mitochondria that can move fast, some portion, some subset will be moving slowly. So these would just be average Doppler numbers for these processes. But they're all basically in the Doppler regime, and so what we're dealing with are actually Doppler edges in the spectroscopy. And again, lots of processes overlapping, and so the Doppler edges will all be essentially superposed on top of each other, and that's going to soften them out. But that's why we're going to be talking about Doppler edges, Doppler knees, and knee frequencies when we do our spectroscopy. All right, so now I am going to switch to the first of what are really the three main topics of this talk. And so the first is going to be biodynamic imaging. And so this is a technique, we've been working on this for a number of years now. And so I'll talk about the origins of BDI and describe how we do Doppler tagging of deep light that's deep inside of tissue. We do coherent detection through holography, and we perform motility contrast imaging and also tissue dynamic spectroscopy. We can even do higher resolution spectroscopy, TDSI, and then also at the very end, I'll just mention briefly some phase 2 human clinical trials that we've completed using biodynamic imaging as a way of looking at properties of cancer. All right, so this is the origin of biodynamic imaging. It really began, I'm going to say, with this paper by Pine and Weitz, the Fissure of Letter around 1988. And so they were shining laser light into turbid media, not living tissue samples back then, but turbid media, so it'd be like milky water. And then you could actually time out the time of flight for the photons through the system. And it's multiple scattering. And so you've got lots of partial waves. And this is the same phenomena that when you put your hand over a flashlight at night and your whole hand glows red. And so the photons from the flashlight, at least the red photons, are just bouncing around. They're not absorbing. But it's high multiple scattering. And so a very wide distribution of optical path lengths for the photons that are transporting through that media. And so you can do various types of spectroscopy with these diffusing waves. And so that was really launched in the 80s. A few years later, another piece of the puzzle was optical coherence tomography. That was invented by Jim Fujimoto, and that was in 1990. Today, this is in ophthalmology clinics. It's a standard technology, so it's highly commercialized now. This is a modern example of a cross-section, an OCT cross-section. So this is laser ranging. So this is basically using laser light like it's radar, and you actually do a radar image or a LiDAR image of your retina. And so these are various layers of the human retina. And so that's using coherence properties of light to do the laser ranging. And so I'll talk more about that in just a minute. So we rely on digital holography. So this was pioneered by Schnars and Jupner also in the early 90s. And that's where they pretty much were just doing standard holography. You'll kind of recognize this is the conventional holography setup over here. But instead of having the photographic emulsion, which is a real pain to work with, they just replaced it with a digital camera. Back then it was CCDs. Now everything is CMOS cameras. And then we came along about 10 years later with holographic optical coherence imaging, where we really kind of combined everything. So we combined the multiple scattering from DWS, the coherence gating with short coherence light sources from OCT, and the coherent detection of digital holography. And then this is one of the very first volumetric images we made of a tumor spheroid, and that was my postdoc Ping Yu back in 2004. So what we want to talk about today is Doppler tagging of deep light. And so this is where we're doing Doppler spectroscopy of light which goes fairly deep into tissue and comes out. So this is a photon Monte Carlo simulation. And so you have a line of photons coming in, and they start to scatter around. And so you can see the multiple scattering. In fact, you actually get what's called a scattering cone out of this. But most of the photons are forward scattering. They are scattering, but it's forward scattering. And the Doppler shift for forward scattering is close to 0. But generally, there'll be 1 or 2 hard backscatter events. And so that's where you pick up the Doppler. And then the photon will forward scatter all the way back through the tissue. And let's say we go a ½ millimeter deep inside this tissue. And so the photon goes in, it gets tagged with the Doppler frequency, it brings that Doppler frequency out, and then we can measure that with interferometry. The coherence gating, this is the laser radar part of it. And the way to think about this is it's just classic radar. Let's say you have a laser pulse, and the pulse shines into this turbid medium. It might be milky water, or let's say we've got some reflectors in there, some partial reflectors. And so there's this distributed time of flight that comes out, but you'll have these coherent reflections. That's these 3 little blips here that are reflecting off these 3 structures. And that's on top of a multiple scattered background. Now, this is not drawn to scale. The background's about 1 million times brighter than these very small coherent reflections that we're trying to pick up. And so it's a severe detection problem, but because these are coherent, we can take a pulse from the same light source that's coherent with that light source, and we just sweep the time of flight, just with moving a retroflecting mirror. And as we sweep that time of flight, every time that these overlap in time at the detector, you'll get interference. And so the interference actually is significantly boosted relative to this background. The background's still brighter, but the background is structureless. The interference will actually produce, in our case, holographic fringes, and so that's what we capture on the camera. And so that's the way that this is demodulation, but what we'll do is actually pick up these coherent reflections coming from inside of tissue, and just by sweeping the delay time, you can actually select the lights that's coming from different depths inside of the tissue. All right, so this is our optical setup. So we call it OCI, or optical coherence imaging, sort of after OCT. But we don't really do tomography. In fact, initially, there was no tomography. We had analog video cameras, and we had actual holographic films. And so there was no tomography at all. It was conventional holography, and we were just having a video camera looking at the holographic readout. Today, that's all in the past. Now everything is digital cameras, and the holographic reconstructions are all digital. But the principles are the same. And so you have a low coherence light source, and that goes through a signal arm to a target, and this, we have a beam splitter bouncing it up to the target here. We also have a reference arm, so this is a Mach-Zehnder configuration with a variable delay stage. And so just by moving these mirrors back, it takes longer for the photons to get to the CCD, which will then interfere with photons that went deeper into the tissue. And so that's how we do the laser ranging. The image is really the interesting part of this. At least I find it the interesting part, because we have a couple of lenses. There are actually 3 lenses here. These are Fourier transform lenses. And so there's a first Fourier transform when you go from the target to a first Fourier plane. There's another Fourier transform that brings you back to the image plane, and then another Fourier transform that brings you onto the CCD, or these days it's a CMOS plane. And so we're doing a Fourier domain holography. And so the reference is coming in at a slightly off angle that writes these interference fringes on the digital camera. But the speckle is actually Fourier domain speckle. It's not image domain. And so therefore we capture that Fourier domain hologram, and then we do a numerical reconstruction to extract the actual cross-sectional image of the target. And so this is the raw hologram. And if you zoom in, you can actually see these stripes. These are the interferometric holographic stripes. And we do a numerical 2-dimensional Fourier transform. And so because this has a spatial carrier frequency, we get 2 sidebands. This was in the Fourier domain, so this is now in the image domain. So the sidebands are actual images. They're the actual cross-sectional image of the tumor at a selected depth inside of a tumor. And so then this is just a blow-up. It just looks like speckle, but this is a tumor spheroid, and so it's an optical cross-section, completely non-destructive, non-invasive. The thickness of this optical section is about 20 microns, because that's the coherence length of our light source. And so it is an optical section, a real-time optical section of the tumor. And then, being a camera, we then just take lots and lots of frames at a frame rate, and we create movies, we have dynamic speckle, and then we can do spectroscopy on that. But before getting into spectroscopy, one of the simplest things you can do on the image reconstruction is just look at the activity of the speckle. And so this is a case where we're going to do laser ranging into a tumor spheroid. This is a really large tumor spheroid. This is about 1 millimeter in size. So we had a collaborator, John Turek, who was able to grow these to these very large sizes. And so here we are at the bottom of the tumor, the coherence gate. That's about a 20 micron section at the bottom. We're pushing that coherence gate up through. This is the midsection. And you'll notice here, so what we're doing is we're color coding on just the activity of the speckle. And you notice this bright red ring around the blue core. And in fact, this tumor is so large, it's avascular. And so it means that there's nutrient depletion and oxygen depletion in the middle. And so it's a hypoxic core, also somewhat necrotic. And so you can see the proliferating shell surrounding that. And then we can push the coherence gate up through to the top. We can stack these together into these reconstructions, and we can do animations on these and fly-throughs and the whole works. But the point is that this was the first time that motion was used as image contrast for volumetric imaging. And so we published this. This was my grad student, Quan Zhang, and we published this in 2007. And so this data is actually somewhat famous. Anyone who's gone in the south entrance, if you look at in the entranceway at the south of this building, you'll notice this on the wall. And so this has now been enshrined in some sense in the building entranceway. And so this is this data from Quan Zhang in 2007. So we've come a long way since then. So this is our current technology. This is an insert, which is so this is a biodynamic insert that goes into an Olympus IX73 microscope. And these are examples. In fact, actually, this is the dynamic speckle. If we just set our coherence gate at a fixed depth inside of the tumor, this is actually what we pick up. This is the movie. And so that we can turn this into our motility contrast imaging, or we can do it volumetrically, in which case this is a cross-section. Here, the blue isn't as pronounced. This is a smaller tumor, so the hypoxia was weaker. But you also do see that there's enhanced blues in there. There's more reds on the outside. And so again, we can pick up that heterogeneity inside the tumor. All right, so we want to do more than just look at how much motion is going on. We'd really like to do Doppler spectroscopy. And we'd also like to track how these tissues respond in time. And so to do that, we're going to talk about time frequency formats, which is really spectrograms. And so this is an example of a bird call, and its frequency is being modulated as a function of time. And so you can break that up into a spectrogram of frequency as a function of time. And so this becomes the voice print of that bird. And we have our own voice prints. In fact, that can be used for security because we're all unique in how our voices sound. So we want to do the same thing using biodynamic imaging and looking at the spectra, the Doppler spectra coming off of living tissue. So I actually have the spectra to the right here. And so these are tumor spheroids. I have frequency along the bottom axis, and we're going across 3 orders of magnitude here from 10 millihertz up to about 10 hertz. And this is the very characteristic spectrum we get. Like if you look at the initial spectrum, that's this red. And so we do get a Doppler edge to it. And there's a roll-off, there's a Nyquist floor, there's something we call the plateau up here. And then after applying a drug to that, so we establish a baseline over about 4 hours. And then in this case, we're applying colchicine. And the colchicine then changes the Doppler spectrum. It changes the spectral weight at these different frequencies. And so what I'm plotting over here is the relative change in frequency. And so this is the baseline, so it's all roughly green, which is 0. We apply the drug, and then we get an enhancement at low frequency, a suppression in mid frequencies, and an enhancement at the high frequency. So this is the fingerprint, the drug fingerprint for colchicine, in this case acting on a colon cancer adenocarcinoma. Or we can look at a canine B-cell lymphoma and a different drug. Colchicine was a cytoskeletal drug. Vincristine is also a cytoskeletal drug, but a different mechanism of action, or MOA. And you can see a very different fingerprint or spectrogram. And so, in fact, we can start to then build up libraries of how different drugs change the different Doppler spectra in different types of tissues. Most of these are canine B-cell lymphomas, although I'll also have a couple colon adenocarcinomas down here. But you can build up these libraries now of characteristic response to these drugs, and you can even go a step farther. And you can start to say, well, okay, if we take biopsies from patients, in this case, canine patients, and this is my collaborator, Mike Childress, who's a vet doctor here on campus, you can actually look at the group of resistant patients or dogs that were resistant to their chemo, and then you can look at the dogs that were sensitive, and they were getting CHOP therapy, which is also what humans get for B-cell lymphoma, and these are the monotherapies that make up CHOP. And you can just notice, we'll take a look at doxorubicin, just look at the striking difference between the average resistant response and the average sensitive response. And so then the idea here is that you can predict. You can take a dog with unknown sensitivity to their chemotherapy, and if their patterns match one or the other, you could actually predict whether they're going to be resistant. If they're going to be resistant, then the doctor can actually choose to do other types of treatments. And so then this is an example of doing a holdout cross-validation on classifiers based on these patterns. And we do get a couple of dogs wrong. There are actually 3 dogs here that we get wrong out of about 20. And so that gives us about an 85% accuracy. And that is typical for this technique. And so out of 20 patients, we'll, let's say, roughly get about 3. 2, 3, or 4 of the patients we might misclassify. But the point is that if we can classify dogs as being resistant, then they can avoid the therapy and go to some other treatment. And so we've taken this from preclinical trial in dogs to clinical trials in humans, all the way up to phase 2 clinical trials. And we've had 4 of these now. So human ovarian cancer, that was with Northwestern, human breast cancer with a group of clinics in Arizona, human bladder with a group of urology clinics on Long Island, and then also human esophageal at IU School of Medicine. And in all these cases, I'm going to say we're kind of hovering in the range of about 85%. So out of 20 patients, we do typically get about maybe 3 wrong. But it also means that we're getting 17 right, that we're calling correctly that they'll be either sensitive or resistant to their therapy. And so across all of these different cancers, it looks like we have an ability to identify when patients are going to be resistant. And so moving forward with sufficient funding, we could actually do this with blinded trials. And then that would be then phase 3 trials where we really prove whether this idea is going to work or not. And so that's still to come. So we still have to get over the barrier of getting into these phase 1 clinical trials. All right, so I do want to just say a little bit more about the spectroscopy. Everything I showed so far were averages over small samples of about a cubic millimeter each. And so this is now a single sample of about a cubic millimeter in volume. And what we can do is we can map out that there are different drug responses in different parts of the tumor. And so this is like one signature in one location. Here's another signature in another location. And so cancer is heterogeneous. It's one of the biggest challenges of trying to treat cancer is that it's not just one disease. Even one biopsy is not one type of material. It's a whole set of materials that could be responding differently to chemotherapy. Some parts of the tumor might respond well. Other parts of the tumor might not respond well. So we can map this out. And so this is actually one of our first attempts at mapping it out using TDSI, or tissue dynamic spectroscopic imaging. And so my student, Li, did a 4-color merge where he took 2 of these biodynamic biomarkers that we can define, and then he made these maps across these samples. These were esophageal cancer samples here. And then we also did a histology comparison working with a group down at Tulane. And so we could match up part of our biodynamic maps with the histology maps. And so that's a way that we can tackle the heterogeneity of cancer. So before I move to the next topic, I do want to say something about the frequency sensitivity of our technique of biodynamic imaging. So I showed you the Mach-Zehnder configuration, but there are other configurations, and this is one of them. This uses a Fresnel biprism. And so it's just this was actually invented by Augustine Fresnel around 1819. And so this is actually a very old technology, but it works really well at producing interference fringes. And so these are it's a very low apex angle. This is actually exaggerated the way it's drawn here. The apex angles can be quite small in the range of like 10 degrees. And so then you can get these interference regions out here in space, and so you'd put your digital camera right here. So currently we would lose the coherence gating, but what we're gaining is incredible stability, because this is what's known as a common path interferometer. And it allowed a visiting exchange student from Columbia, who's an undergraduate student, who actually pushed our frequency sensitivity from 10 millihertz actually down to 100 microhertz. And if you do that simple conversion that I talked about earlier, that's about 30 picometers per second, or about 1 hydrogen atom per second is the bottom velocity that we could actually pick up with this. That's the stability. And you can actually see that's where the 1/f noise of a static target, that's white paper. But at 100 microhertz is where the 1/f crosses over, and so that would set the lower limit for what we can detect. So our relative sensitivity is 100 microhertz over the center frequency of our light source, which in our case is 3.6 10 to the 14. So we have a sensitivity of 3 times 10 to the minus 19. And I want to point out that we had a speaker from NIST give a colloquium about a month ago. He has one of the best atomic clocks on the planet, and his frequency stability for his atomic clock is actually 6 to the minus 19. And so, in a sense, we're a factor of 2 better than the best atomic clock. The atomic clock costs about $10 million. Our unit costs about $10,000. And so there's about a factor of 1,000 difference there. LIGO is another factor of 100 even better than the atomic clocks. That actually gets down to 10 to the minus 21. And so you can argue that this is the most sensitive measurement ever made by mankind. Now, I do want to point out that while we're working with $10,000 equipment and NIST is working with $10 million equipment, their frequency sensitivity or stability is absolute. Ours is just relative. It's really looking at the relative frequency on, let's say, one side of this Fresnel biprism relative to the other. And so this is actually easy to do with simple optics. What the atomic clock people do and what LIGO do, those are hero experiments. So we're not actually competing against them because we're looking at relative changes. But still, we're looking at relative changes down to 3 times 10 to the minus 19. And so that's actually pretty impressive for something you can just throw onto a benchtop in the laboratory. Coming now to the second topic out of 3 that I'll be talking about, it's phase-sensitive biodynamic imaging. And so everything I showed so far was actually under homodyne conditions. The detection is heterodyne. We are doing holography, and so there is a reference wave, and so it's holography. But we were throwing away the phase and just looking at amplitudes. But because we're doing holography, when we do that Fourier transform to reconstruct our image, we get real and imaginary parts of the image in the image plane, then we can actually extract phase. And if we do post-processing phase stabilization, we can even study the properties of that phase. And so I'll talk a little bit about the phaser plane, how we get speckle phase, and then I'll say a little bit about black swans. And so black swans are these outlier events. And so it has to do with statistics where you get rare but high amplitude events that can skew statistics. And those are generally called Levy statistics. And it looks like there may be Levy processes going on inside a living tissue, and I'll give a little bit of evidence for that. So this is one of my favorite experiments that one of my students did. He actually did it on his own. He thought this up and was so kind of thrilled that he actually kept it a secret from me. And so he worked on this for about a month and then just showed up one day at the weekly group meeting and he gave a full presentation, all accomplished and done. What had happened is I had challenged him. I said, well, look, we're doing Doppler spectroscopy of these very amorphous, very broadband processes in living tissue. Wouldn't it be nice if we could actually calibrate that to actual Doppler signals? And so what we need is to get Doppler speeds that are comparable to the kinds of Doppler speeds that are going on inside a living tissue. And so those are pretty small. Those are hundreds of nanometers per second. How do you calibrate that? And so he came up with this great idea. We work with these 96 well plates, and they have growth medium or water in these wells that are containing the samples. But the water evaporates. And in fact, it's kind of a problem. When we do very long experiments, we have to make sure that the water doesn't evaporate to the point where the target gets uncovered. So he realized that, well, okay, the top of the water is actually going down over hours. And so he could use that as a calibrated reference. And so to create his target, he actually put a piece of toilet paper floating on the surface of the water in this 96 well plate. And then the light's coming up, it's scattering off of this target, the water's evaporating, and so this target is moving down. And for instance, it's moving down typically with 100 nanometers per second, which was giving a Doppler frequency of 150 millihertz. And he was even able to then do phase extraction. He stabilized the phase, so these are now looking at the real and imaginary parts of the phase. And he changed temperature. And so as you change the temperature, you change the evaporation rate, and you change the speed, and here you have the Doppler peak, right? So here's the Doppler peak, the heterodyne spectrum over here, and you get the classic Doppler peak, which is going faster at higher temperatures, okay? And so then this was getting up to about, in this case, 0.29 hertz, and in the lowest case, it was 150 hertz. Okay, so this was a great way of doing the calibration. Also a demonstration that we could do phase-sensitive BDI, And so then that is actually where we can now do phase retrieval. And so when we do our reconstruction, I mentioned that we have the spatial carrier frequency. So when we do the Fourier transform into the image domain, we actually get the two sidebands. These are now image domain cross sections of a tumor spheroid. So here's intensity, here's the phase, and then a blow up of the intensity, blow up of the phase. So you can see the phase is going between minus pi and pi. And there are actually what are called optical vortices in here. There are singular points where the phase wraps around. And if it wraps around one way, it actually has what's called a churn number. And so it can have a churn number of +1 or a -1 or +2 if it wraps twice, or -2. And so this is topological optics. And so when you're doing speckle, there are singular optical points in there that have topological constants associated with them. And as you all know, topological physics is really hot right now. And so there are groups that study that. We actually are not currently studying that, but the point is it's just an intrinsic part of our data set. So what we're tracking is the fact that the electric field is undergoing a random walk on the phasor plane. Now this is a completely different random walk from what's going on inside the living tissue. In the living tissue, you have this active transport that is doing the random walk with long persistence. And then when we're doing our reconstruction and we're looking at our electric fields and they have real and imaginary parts, there's a phasor. So a pixel will have a phasor value and it's going to be performing a random walk on the phasor plane because of the dynamics inside of the tissue. But it's not a one-to-one. There's a transformation of the random walk motion in the tissue to the random walk phasor on the phasor plane. And so it's not one-to-one. We have to actually understand what that transformation is. But the point is that we can set an origin, and then as the phasor undergoes its random walk on the complex plane, we can track the delta phase, and then we can histogram it. So this is a histogram of delta phase, in this case stationary paper, and this is also referenced at an early time and then to later times. And so there's a little bit of phase diffusion, or actually dephasing or decoherence, if you will, over time, but actually pretty good. I mean, so not very strong, and so we actually get this nice Gaussian distribution of phase steps on the phasor plane. And so that was for a static target. Now, what was really intriguing is that we looked at one of our living tissue targets. So this is a tumor spheroid, probably, I don't have it here, but it's probably a DLD. And we get a spectrum, or it's a PDF, right? It's a probability distribution function of delta phase in radians. And we get a knee, it rolls off. So again, this is not spectroscopy. Even though this looks a lot like our spectra, it's not. This is a PDF, not a spectrum. But if we try to fit a Gaussian to it, it's a terrible fit. And so we might also fit a Cauchy, or what's known as a Lorentzian. Lorentzian, there are reasons to believe that it should have been Lorentzian, but it's not. It's something in between. And you don't have to look too far before you run into something called Levy statistics, right? So you can have Cauchy statistics, you can have Gaussian statistics, but there's actually a continuum of statistics between these two called Levy statistics. In fact, there's something known as Levy alpha, where it's the characteristic function has a stretched exponential. And so what you get is, in the one limit of when alpha is 1, you get Lorentzian or Cauchy. And when alpha is 2, you actually get your Gaussian. But you can just tune it continuously, alpha from 2 to 1, and you can get lots of different scaling behaviors, all under Levy statistics. It's stable. And just as if you add a Gaussian distribution to a Gaussian distribution, you get a Gaussian distribution. Everyone who does error analysis knows that. Same thing's true of Levy. If you add a Levy distribution to a Levy distribution, you get another Levy distribution. And so it's what's known as a stable, Levy stable distribution. And so the black line here is the Levy fit to the PDF. And in fact, we can extract a Levy alpha parameter here of about in the range of about 1.5. And so then that's really intriguing, right? So that actually brings up the idea that perhaps what we're seeing are Levy processes in the living tissue. And Levy processes have been found in using live cell imaging, and also just in animal biology. It turns out animal foraging is like Levy. And Levy is characterized by local clusters where it looks like kind of Brownian, and then you do these long flights. And it's easy to see why this would be good for foraging, because you might actually deplete all the food here, and so then you do this long flight, brings you to a new area, you deplete that, and then another long flight. This is a Levy with an alpha of 1.5. So this is Cauchy that has these really distinct long distance flights. This is an alpha of 1.5. This is Gaussian. Okay, so that's your classic Brownian or random walk that everyone knows about. So the Levy 1.4, it's like it's Brownian in these regions, but then you have these long flights. And so cell movements have been, in some cases, seen to be have Levy distributions. Also, molecular motors can have Levy distributions. And so we wanted to take a closer look at that. So here's stationary paper. And so now we're doing a Levy fit. We're also doing Gaussian and Cauchy, but we're doing a Levy fit. 1.9, the fitting is not that accurate, I'd say. I mean, the alpha is probably plus or minus 0.1 or 0.2, but the point is that this is pretty close to Gaussian. This was the vertically moving toilet paper. He got 1.8, but still, it's pretty close to Gaussian. And then this is living tissue, 1.65, and so in fact, that's actually quite a bit smaller. And then when you have an infection, the alpha gets even smaller. And so what that means is we're getting a longer tail. And that is with an infection, in this case, with, I believe, E. coli. And so we've also looked at infection with the Listeria and Salmonella. And so the bacterial infections are actually changing the alpha parameter. And the alpha parameter is a fundamental. It's just like the standard deviation of a Gaussian. The alpha is a fundamental property of a Levy distribution. And so I'm going to switch now to the third and final topic. I'm going to go through this fairly quickly because I'm approaching my time now. But this is the idea of a sentinel tissue. And so the sentinel effect in living tissue. And what we're doing is we're letting tissue be infected by a pathogen, by bacteria and also by viruses, where the early stage infection modifies the behavior of the tissue before there's sufficient numbers of pathogens to scatter light off of directly in the case of bacteria. In the case of viruses basically don't scatter light. They're too small to scatter light. But the viruses modify the behavior of their host tissue, and so that becomes a sentinel. It's a living sentinel that is a early warning system for the infection. And so we'll actually show you how that works. And so this is the idea of the canary in the coal mine. And so here you've got your canary, a dead canary. This had to do with methane, or was it carbon dioxide? I actually forget now. But if it's the gas that can be dangerous to the coal miners, the canary dies because it's very sensitive. And so that is a living sentinel that tells the coal miners to leave the coal mine. All right, so we're going to use the same idea. And so in this case, this is work by Dawith Lim, who is actually just defended, will defend next week, actually, his PhD thesis. So this is an example where it's infected with E. coli, Salmonella, and Listeria. So you establish a baseline. This is frequency. These are actually arbitrary units down here, but this is frequency 3 orders of magnitude. And then this is time going up to 25 hours. And so then what you do is you infect with the E. coli after establishing the baseline. And then about 3 hours later, you start to pick up your initial sentinel response of how the tissue is responding to this very early infection by the bacteria. And that'll go on for about 9 hours before the bacteria actually get so plentiful that they start to scatter light directly. And so most of what you're seeing up in this area here is direct light scattering by the bacteria. But there is this thing that we call the sentinel window. It's where we're detecting the change in the dynamics of the host tissue before we can really detect the presence of the bacteria directly. And so then we have the same effect for Salmonella and also for Listeria. And these are very dramatic changes. These actually, these relative changes go from minus 1.5 to plus 1.5. So very large changes caused by the bacterial infections, much larger than we saw with any of the drug responses that we looked at earlier. Now, we also wanted to do this for viruses. As I mentioned, viruses, they don't scatter light. Even when they're highly plentiful, they're not going to be scattering light directly, but they will be influencing the health of the tissue. And so this is work by Shivani Mahajan. And here we have a control sample. This is over even longer times. This is going out to about 48 hours. This is an adenovirus 6, which is infecting a DLD1. And then we subtract the control from this. And so this is the viral signal. And there's this early signature here. And so we get this early signature of the viral infection prior to sort of massive collapse of the tissue because of the pathogenic properties of the tissue. Also, the frequency collapse, the spectrum collapse isn't nearly as pronounced as it was for the bacteria, but still measurable, and we can detect it early. All right, and so I'm just going to sum up the talk now. And so how far have we come? Well, we did the first volumetric motility contrast images. And so we were using living motion as our image contrast, and we were doing that with the full volumetric imaging in these large tissue samples up to about 1 millimeter. We're performing Doppler spectroscopy of intracellular transport, and we can correlate the Doppler spectra with the treatment outcomes of patients. And so then the hope is that we can start to do therapy selection at some time in the future. And then also I demonstrated that we're looking at these tissue sentinels for fast infection assays. So where do we go next? Well, we're still working on anomalous transport. We really want to nail down sort of the smoking gun for Levy transport inside a living tissue. And so we're working on that. The very low frequency results using the Fresnel biprism, that actually opens up an opportunity to look at metastatic cell crawling. And so we're going to be looking at that, digging a little bit deeper into tumor heterogeneity. Also, as I said, ideally we'll get to therapy selection for cancer patients. Also, we're looking for an organelle Doppler edge. If we have a very high-speed camera and enough photons, we should be able to actually see a Doppler edge to organelle motion directly. Currently, we mostly pick that up as a Nyquist floor, but we'd like to see that edge directly. And so acknowledgments, I've had many years of very good students, lots of funding sources, a few postdocs. I have not had a lot of postdocs, but Ping Yu was one of the most important ones. He's the one who really helped launch biodynamic imaging. Lots of collaborators around the world, collaborators at Purdue. And so thank you. And I can't end without saying that if you're interested in some of the history behind the interferometry that I talked about today, especially, let's say, Fresnel and Mach and Zender, then I have a book at Oxford University Press called Interference, and it's the history of optical interferometry. And so thank you.